So good morning students. We'll continue our discussion about restriction endonucleases. In the last class we saw many aspects of restriction endonucleases. We discussed their role in bacteria, how restriction endonucleases are named, and also the definition of restriction endonucleases. Now let us today discuss about the action of restriction endonucleases. That is, how restriction endonucleases act on DNA. As we have already discussed in the definition, that restriction endonucleases have the special ability to cleave DNA at specific points, to cut DNA at specific points. So today let us continue with its action. Let us take the example of the enzyme EcoR1. This is the enzyme which we discussed yesterday also, from Escherichia coli. One of the very important, very familiar, very common restriction endonuclease which is used in genetic engineering. And as we discussed yesterday, the enzyme EcoR1 acts on the sequence GAA-TTC, isn't it? So this is 5' end, this is 3' end, and if we go from here again, it is GAA-TTC, palindromic sequence. As we know, these complementary nucleotides are held together by hydrogen bonds, and this is the point where the DNA is cut by the restriction endonuclease. So as we have seen, restriction endonuclease acts on double stranded DNA. Alright, so when this sequence is present with a DNA strand, imagine this is a big DNA strand, and in between this sequence is present. So when you add EcoR1 to this, the enzyme EcoR1, the restriction endonuclease is added, what happens is EcoR1 is able to specifically recognize this sequence, bind to this sequence, and then cut the phosphodiester bond between G and A. It cuts this phosphodiester bond between G and A. So all these nucleotides are held together by phosphodiester bonds. We know this already, right? But the enzyme breaks this bond specifically. So what happens is, actually the DNA gets cut like this. Alright, the DNA gets cut like this. So ultimately you get two pieces of DNA, two segments of DNA which go like this. This is one, and this, because there's a cut here and here, the other strand comes out like this. The other segment comes out like this. So you get two such sequences. Alright, now if you see these two ends, okay, what do you see here? This one of the DNA strands is short, the other one is long. This is long, this is short here. Okay, so these are called as overhangs. They are called as overhangs because you can see that it's kind of hanging, isn't it? So they are called as overhangs. So such ends are also called as blunt ends. Sorry, I'm so sorry about that. Such ends are called as sticky ends. Why are they called sticky ends? Because if you bring this and this together, you can still see that they are complementary to each other. They are still complementary to each other. If they come together, they have the ability to form hydrogen bonds, and they have the ability to stick with each other. So they are simply called as sticky ends. Alright. Let us take another example. So this is one of the first enzymes that we discussed. Let us take another example by the name ALU1. ALU1 is another restriction enzyme which is from the bacteria Arthrobacter luteus, and ALU1 recognizes the sequence A, G, C, T. Only 4 base pairs are there. A, G, C, T. If you go from this side, it is again A, G, C and T. Every time we write it from 5' to 3', and you can see this is also a palindromic sequence. Now ALU1 recognizes this sequence AGCT. So when this DNA is taken and ALU1 is added, what happens is ALU1 cleaves the DNA at this point. Okay, so what happens is that these two phosphodiester bonds are cut and you get 2 pieces of DNA like this. Like this. Okay, so these don't have those overhangs like these do. You can see that these ends have the overhangs, right? Like this. You can draw it like this also, showing the overhangs, but these don't have those overhangs. So these ends are called as blunt ends. Okay, they are called as blunt ends or flush ends. These are called as sticky ends or cohesive ends. Okay, so these are 2 ways, different ways in which different kinds of ends are produced: blunt ends and sticky ends. Which one do you think will be better for a genetic engineer if he wants to later on join the 2 pieces of DNA? Obviously this is going to be easier because they already have the sticky ends and they can come together and form hydrogen bonds, and it will be easier to stick them back together. So sticky ends will be easier. So EcoR1 is an example of an enzyme which produces sticky ends. ALU1 is an example of an enzyme which produces blunt ends. So when are sticky ends produced? When are blunt ends produced? Sticky ends are produced when the enzyme makes a cut in different places on the DNA, on the 2 DNA strands, isn't it? So you can see these 2 DNA strands in different places the cuts are made, whereas in this case cut is made at the same place. So makes a cut at the same point on the 2 ends of of the 2 DNA strands. Alright, so this is how sticky ends and blunt ends are produced. Or we can also say sticky ends are produced when restriction enzyme cuts the DNA strands away from axis of symmetry. Blunt ends are produced when a cut is made at the axis of symmetry. Alright, for example, AGCT, TCGA. If you see, this is the axis of symmetry. Okay, these two are symmetrical to each other. So when the cut is made at the axis of symmetry, you get blunt ends. Now for GAATTC, the EcoR1 site, where is the axis of symmetry? Axis of symmetry is here, but the cut is made at this point. So when the cut is made away from the axis of symmetry, you get sticky ends. When the cut is made at the axis of symmetry, blunt ends are obtained. Okay? So this is about the action of restriction endonucleases and the types of ends they produce. So which is better among the two? Sticky ends are better than blunt ends. I've already told you the reason why. Because they have ends which are complementary to each other and which can form hydrogen bonds with each other. Therefore, they can be brought together much easily. Finally, let us discuss the role of restriction endonucleases in genetic engineering. In genetic engineering or RDT, what is the role of restriction endonucleases? Number one, they used to cut the vector molecule precisely at a specific point. So we've already discussed about vectors, how they are used to carry the gene of interest. If you have a vector like this, and you want to cut open the vectors, so you can use restriction endonucleases, and what will happen, the vector, because restriction enzymes are very specific, the vector gets cut every single time. It gets cut at the same position. So precisely at a specific position or a point, the vector gets cut. And secondly, it is also used to isolate the gene of interest. So if you have a large genome, supposing you have the human genome, alright, this is a human genome, very large DNA, and your DNA is some gene of interest is somewhere here. You want to specifically cut it open and you want to remove it. So what can you do? You can add restriction endonucleases to it. The genomic DNA. So when you add the restriction endonucleases to the genomic DNA, it gets broken down into a number of pieces, because wherever it has its specific sequence which it recognizes, it goes and cuts the DNA. So you get a number of fragments of DNA, and in one of the fragments of DNA, your gene of interest will be present. So then you can go about isolating only this specific gene of interest, leaving all the others. So therefore, restriction endonucleases are used to isolate the gene of interest. These are the two main roles of restriction endonucleases in RDT: to cut the vector molecule at specific points, and it is used to isolate the gene of interest. Another technique which uses the specificity of restriction endonucleases is known as RFLP, or restriction fragment length polymorphism. Okay, so since it is connected to restriction endonucleases, we will be discussing it here. It is called as RFLP, and it stands for restriction fragment length polymorphism. RFLP. Alright, now let us first try to understand what this restriction fragment length polymorphism means. Now you see, among humans also, though we all belong to the same species, our DNA is unique. Even if you take two very closely related individuals, let us say A and B, alright, let's say they are very closely related individuals, maybe brother and sister or cousins, alright. So this is individual A and individual B. If you take their genomic DNA, if you isolate their genomic DNA, alright, and you subject it to restriction endonucleases, the same type of restriction endonucleases, alright, what is going to happen? The DNA is going to get fragmented, right? There you will get numerous number of fragments of the DNA. Now, if you are able to separate these two fragments on a gel and see, there are techniques to separate the fragments of the DNA that are obtained depending on their size, you will see that no two individuals will get the same pattern of the DNA fragments. This is called as RFLP. No two fragments, though they may be closely related like these two individuals A and B, their pattern, the restriction fragments that we get from their DNA and the pattern that we get from these restriction fragments will not be similar. You can see here that A and individual A has got 3 different fragments, whereas individual B has got only 2 fragments, and that too, this one is totally different. This is a totally new fragment. Alright, so every individual gets a different pattern like this, leading to what we can call as a DNA fingerprint. Alright, just like how we have, each of us have a different fingerprint on our fingers, like that our DNA also has this kind of a fingerprint with respect to the restriction sites that the DNA has, and this gives us what is called as DNA fingerprint. Now look at the name of this technique, restriction fragment length polymorphism. You are using restriction enzymes, so therefore the term restriction. You are taking the fragments that are obtained by doing a restriction digestion, so that is why this is fragment length. The length of these fragments is going to be different. That is why the term polymorphism has been used. Polymorphism actually means many different kind of patterns. Okay, that is the meaning of that. This technique, RFLP, is mainly used for this technique called as DNA fingerprinting. I'm sure you have heard of the technique called DNA fingerprinting, which is used to find criminals, which is used to find criminals using even a little bit of DNA found in the crime site. So the basis of this technique, DNA fingerprinting, is nothing but RFLP, which is very much based on the restriction endonucleases that we have seen. Okay, I hope this is clear. We will stop with this for the day.